today's speaker for the Star Trek High Fidelity Seminar Series, Dr. Madhu Lal-Nag. After completing a bachelor's degree in microbiology and biochemistry at St. Xavier's College in Mumbai, India, Madhu attended graduate school, earning a Master of Science degree in biochemistry from the University of Mumbai in Mumbai, India, and a Master of Science degree in biochemistry, a Master of Business and Science degree from the Keck Graduate Institute of Applied Biosciences in Claremont, California. She then earned a Ph.D. in molecular and cellular oncology from the George Washington University in Washington, D.C. Madhu carried out postdoctoral fellowships at the NIH National Institute on Aging in the Laboratory of Molecular Biology and Immunology, and at the NIH National Center for Advancing Translational Sciences, or NCATS, in the Division of Preclinical Innovation. Her work at NCATS focused on the development of single and multi-cell type tumor spheroids and organoids for high-throughput small molecule and functional genomic screening. In 2015, Madhu accepted the position of Director of the Trans-NIH RNAi Screening Facility, overseeing high-throughput functional genomic screens for the entire NIH intramural program, serving 27 institutes. Her group established an arrayed functional genomics robotics platform to run high-throughput phenotypic screens and lead the development of normal and disease-specific physiologically relevant toxicological models to complement mechanism of action screens. In December of 2018, Madhu moved to the Center for Drug Evaluation and Research, or CDER, at the FDA, where she serves as the Program Director for the Research Governance Council, an advisory council overseeing CDER research. At CDER, she also serves as a scientific advisory board member for the National Center of Toxicological Research and works with a team of pharmacologists and toxicologists to identify areas of unmet need in safety pharmacology. To unite researchers throughout the world in developing and adopting standards and reference materials for 3D tumor models, Madhu established a 3D tumor model standards working group, including stakeholders in government, academia, and industry, who are focusing on the standardization and advancement of 3D in vitro model systems that correlate with patient responses. Madhu also serves as an associate editor of the NIH Assay Guidance Manual, a unique ebook with more than 50 chapters that focuses on assay development for early-stage drug discovery. With that, I will welcome Madhu to begin her presentation, and just a friendly reminder to everyone to please mute your audio connections unless you are speaking.
Thank you, Nate. I really appreciate that introduction. And without further ado, I will start sharing my screen. Please let me know when you can see it. Great. Can you see my screen?
It looks great.
Perfect. Well, thank you, everyone. It's a pleasure to be here today to talk to you a little bit about not so much work, but a mindset that I've sort of been very fortunate to be a part of, from the beginning of my research career up until now, and sort of the lessons that I've learned along the way that have led to insights into the development and utility of physiologically relevant models in drug discovery. I will begin with my conflict of interest statement. The views that are expressed here in this presentation are my own and do not reflect the opinions and views of the US FDA. So there's a lot to cover in terms of, both the science, but also sort of the conversation that I'm hoping to have with this group today. So I'd like to begin by talking to you about the organizational, the layout, if you will, of my presentation. So I'm actually starting with the destination, and I'll tell you a little bit more about why. I think, very often we hypothetically know that, the recipient of all of this therapeutic development is the patient. But I think it behooves us as a community to be able to keep this in mind right from the beginning, when we were investing all of this time and all of our resources in discovery research, to keep the patient in mind throughout along the way till you actually get to the destination. So I'm going to start sort of retrospectively, talking about the patient, talking about the unmet needs in the patient population, and then move on to a trip advisory of sorts, which is nothing more than a checklist, things that you have to keep in mind when traveling to a particular destination. And then I'll sort of conclude with a journey that's actually now going on 8 years that started off at NCATS, and there's some regulatory interest along the way, and is now actually being pushed into human studies. And I'll conclude very briefly with a vision for the future and the importance of team science and innovation. And if there's anything you take away from this today, I'd really like for it to be that collaboration is a team sport, especially when it comes to therapeutic development. There's absolutely no way that any one of us can do this in silo. It requires different perspectives. It requires complementary disciplines to all come together with important but diverse perspectives so that the best ideas are put forward for therapeutic development. So that being said, let's start with destination patient. Now, what do I mean by that? When I was at the National Center for Advancing Translational Sciences, we went through a phase where we talked a lot about what translational science actually meant. You have basic research, applied research, translational research, regulatory science research, and they are all sort of nested or built on top of each other, if you will. When it comes to translational science, I think the most important thing, which is what I sort of said at the outset, is keeping the patient in mind throughout the process of drug development. So this means from the very beginning, when you're designing those complex in vitro assays, or even the more simplistic assays, the diversity and the heterogeneity that we see in the patient population has to be kept in mind and mirrored in those assays, so that as we move throughout the platform of drug development, we have mirrored the patient heterogeneity throughout the process and made it a much more iterative process rather than a beginning through an end. And what do I mean by that? I think what we ended up sort of looking at as a community of people who were screening not only small molecules, but larger molecules, and also RNAi and CRISPR. The biggest and the most important was to understand what your biological question of interest was and how that related to your patient. It's very easy to say, I want to look at XYZ in a nucleus, cytoplasmic to nuclear translocation, plasma membrane integrity. That's all well and good, but how does that phenotype compare with the disease pathology? So it actually involved a lot of us sort of getting together to think about what was the particular question about a particular biology that we wanted to ask, and try and mirror that in a plate. Develop those simple phenotypes and the complex phenotypes. Understand which proteogenins we were going to use to identify these targets and interrogate gene function. And in fact, for a lot of assays that we sort of started off at the time, we ended up doing small molecule screening as well as functional genomic screening to be really, to really help sort of delineate the polypharmacology that was associated with a lot of drugs. And then, the screening is done with a high throughput or a medium throughput system, and we're going to talk about, the pros and cons of each. And of course, there was a very rigorous informatics process that went into selection for primary and confirmatory screens, and then validation assays. So having sort of established a mindset or a paradigm for our screening platform, the next question that we asked was, why 3D? Why are we interested in building this functionality for the center? And really, when it comes down to it, if we're talking about a disease, as it turns out, human beings are not two-dimensional. So again, it goes back to, what do you want to mirror? If you want to mirror human disease in a plate, you can't really mirror it in flat biology. You really have to build in the complexity that you see in both normal human physiology, but also in human pathology. So having sort of understood the why of why we wanted to develop more complex in vitro models, we then sort of sat and thought about, as a community, and not just a community of screeners, a community of chemists, a community of biologists, a community of informatics scientists. We all had to sort of sit together and think about, what are the potential roadblocks? What do we have to keep in mind while thinking about developing more physiologically relevant assays? So I use the word physiologically relevant systems here, but there are very broad terms like microphysiological systems being used in the community. Some people use complex in vitro models, some people use 3D organoids. I think if you were to look at a Venn diagram of all of these different terms, if you will, there's a lot of overlap. And I think it goes, the community at large hasn't necessarily yet decided on very stringent terms and what they encompass. But this paradigm platform over here actually gives you a sense of how, or the thinking behind using different terminology. So we know at the end of the day how interconnected systems work, both in normal physiology as well as in disease pathology. And the idea really is, how do we get to mirror these inter-organ interactions within a dish in an in vitro setting, right? And I think the important thing to remember here is why we use in vitro systems. As we you can't discard one system in favor of another. It becomes incumbent on us as a community to understand the answers that each particular system can actually give you. So if you know what a system can deliver for you, you won't ask the wrong answers of it. And the data that you actually get from experimentation with that system will be valid and valuable. So for example, with 2D cultures, there are many biological questions about mechanism of action within a single cellular population that you can actually, that are actually valid. Then you have 3D organoids and spheroids. And again, the community sort of split with the, in terms of the description, and we won't go into that too much today in terms of the difference. But what I'd like to communicate to you is that whether you're looking at spheroids or organoids, it's telling you about the interactions due to geometry, due to intercellular interactions and intracellular interactions that you don't get in two-dimensional cultures. It can also inform of certain pH and pathophysiological gradients. When you move into what are called tissue on a chip or microphysiological systems, you're now getting the whole concept of flow and tension into a layered three-dimensional multicellular model. So there again, you get sort of the external influences on cellular architecture that you would see in an in vivo system. So once again here, I think what it becomes important for us to know is that in an in vitro system, you can control experimental conditions, thereby really getting to understand the mechanistic understanding of each one of these systems and how they contribute to sort of our understanding of physiology as a whole. To be honest, all models are wrong. No truer statement was ever made, but some models are very useful. So as we go into, as we get excited about developing more complexity into our models, I think it behooves us to understand that all throughput is going to be, we're going to have to sacrifice throughput. And that's absolutely fine because you don't need throughput for every readout. Generally, as you increase complexity, when you go from spheroids to organoids to printed tissues and then flow systems, you will lose throughput capacity because at this point, it's not just about biology, it's about chemistry and the physics of the plate as well. In those tiny microwells, it is extremely difficult to recapitulate 3D human biology as you would expect to see it in vivo. So what does drug discovery in the third dimension really look like? What do we have to sort of really look at in terms of a framework to put assays together? So if you took, for example, the increasing complexity from spheroids to organoids to printed tissues, right? Every one of these systems is going to give you a different answer to different parts of the same question for the same disease that you're looking to perturb. But what becomes extremely important, even before you start asking those questions, really is the assay standardized? There is, I can't stress enough or emphasize enough how important standardization for wild and disease type models really are, because without establishing the robustness and reproducibility of an assay, the data that comes off of that assay becomes useless to be, for anyone to use, not only in the context of that biology, but in the context of any other question when you want to build another iterative model. So the time that you spend in standardizing an assay becomes extremely important. Something else that I want to bring up that we as a screening community sort of came to terms with was that a lot of phenotypes are not necessarily quantitative. So if you wanted to look at nuclear laminar ruffling, for example, in 3D, there it becomes extremely complex. So one has to develop metrics that are accepted across the board for some phenotypes, where you have a combination of both a quantitative readout as well as a qualitative readout in microbial tissues. And then the last sort of qualifier that we sort of spoke about as a community was the importance of pharmacological benchmarking. We do know we have a great inbuilt test in compounds or drugs, really, not compounds, but drugs that have worked in the clinic. Using the drugs that have worked in a clinic for a particular disease type, the idea really was to work retrospectively and test those drugs in these physiologically relevant models to see which models responded like you would expect an extrapolated in vivo response to look like, and which ones didn't. And for those that didn't, what needed to be tweaked to be able to get an in vivo response. So it really is a question of using information that we have both for drugs as well as for adverse effects, and then retrospectively building that into models. So what do I mean by retrospectively building in adverse events? When it comes to looking at toxicology and safety and efficacy as a screening group, as an academic group, the focus truly really is on looking at efficacy. Does the drug engage the target? What is the efficacy? What is the amelioration of the phenotype? But very often we forget to look at safety in this very, very early stage. So investigative toxicology is a field of research, which is sort of like a sister to regulatory toxicology, if you will, where you can really build in this idea of looking at a therapeutic window very early on in discovery, so that you're able to de-risk compounds moving further on. It's all well and good to use extremely complicated models and showing incredible efficacy. And then the moment you go into safety studies, the compound has a terrible safety profile. So one of the sort of mindsets has been to push investigative toxicology into the safety, into the efficacy discovery space, to really see if we can embrace these new technologies like microfluidic models, like PBPK modeling, to be able to evaluate compounds that will go ahead from hit to lead, that have very good efficacy profiles, but have equally good safety profiles as well. One of the major goals of investigative toxicology, obviously, is to improve preclinical decisions. So I'm going to use an example here about what we're looking at in building a model, and this will become relevant with sort of the journey that we talk about in building solid tumor as well as sort of models for metastasis. So if you look at this picture on the left, it is a tumor organoid which has a quiescent zone, a necrotic zone, and a proliferating zone. And when you actually hit it with an anti-proliferative agent, what it's attacking is only the proliferating zone for the most part. What we don't see is what the effect is on the quiescent zone and necrotic zone. And this is going to sort of speak to the importance of looking at multiple parameters in all of our assays, especially when building complexity into those assays. And the reason I mentioned this particular sort of issue in organoid biology is because there are cells here that are untouched by the compound of interest that in theory can then develop a stem-like phenotype and actually repopulate the tumor. And therein, you're looking at the equivalent in a human being of a relapse. So there are some very important things that you have to think about in terms of the disease itself when developing models, so that things like the pathophysiological gradients, things that like the pH gradient, like the oxygen gradient, like the nutrient gradient that you would see in a tumor, that too is taken into consideration when building out the model. So in terms of our checklist or trip advisory, before we actually tackled a biological question of interest, what are the three most important things that we thought, you always sort of have to keep in mind, right? Because all models are different, but there are underlying things that are very that are normalized across the board. So one of the most important things is context of use. You're developing an assay. Do you know what question you are asking? And does the readout actually give you an answer to a question? Standardization, standardization, standardization. I can't emphasize this enough, because we don't have any rules in place for the development of standards and the normalization of data, even from lab to lab, leave alone from institute to institute. So this becomes extremely important when trying to compare data across the board and then make that extrapolation from an in vitro readout to an in vivo result. And then the most important thing, of course, was what we started off with, what is the biological question of interest? And are we really mirroring the biology of a human being in a dish? So having sort of established that these are sort of the most important things that, and by no means is it exhaustive, this was more just, let's keep this in mind, let's understand these three tenets of assay development for microphysiological models, and then learn along the way as we start developing models for disease biology that we're interested in. And that brings me to the journey. This audience needs no introduction to the fact that, cancer is extremely insidious. And not only is each cancer such a different beast from the other one, but even within the same type of cancer, every stage of cancer is actually a different disease. When you think about developing therapeutics for stage 1 or stage 2, where you still haven't breached the integrity of the basement membrane, the mindset or the thought process that goes into that needs to be very different from when you're talking about hitting metastatic tumors, because metastatic disease is a completely different beast. And the burden of this disease really is that as a community, we've tackled the genomic profile of cancers. We're looking at, resistance across patients. But what we haven't really done has been to delineate the different stages of cancer within a patient and model the heterogeneity of the patient population in the in vitro system. So that's sort of the focus that I'm going to bring to the rest of this talk, which is increasing diversity in the in vitro system, so that the compounds that you get and then you push through to therapeutic development really do have a good chance of succeeding in a larger part of the population. And the fact that the mindset for development of therapeutics for solid tumors, hematological tumors, and metastatic tumors are all extremely different. As this audience knows, the disease burden is still tremendous, and much needs to be done in the management of cancer, though we have made huge progress. I'll be talking today about ovarian cancer, which is a which is the leading cause of death from gynecological malignancies worldwide. And not only am I just talking about ovarian cancer, I'm talking about high-grade serous ovarian cancer, which is just one of the ovarian cancer subtypes, which is, really has no clinical management that has a good prognosis. One of the issues with this, obviously, is that for most early stages of ovarian cancer, there are no distinguishable obvious symptoms. So you have a dire prognosis because you're being diagnosed late. Obviously, for ovarian cancer, we have a huge risk profile with respect to genetics, as well as with some women, postmenopausal hormone replacement therapy also is can contribute to high-grade serous ovarian cancer. So, since we really haven't moved the needle in mortality and morbidity for ovarian cancer, a mechanistic understanding of both the tumor and how it interacts in the patient milieu, as well as its microenvironment, is extremely important for us to be able to develop unconventional, innovative, targeted therapeutics. What do I mean by understanding the microenvironment? So if you look on your right here, you have two epithelial ovarian cancer cells that are interacting with the omentum which is multicellular or multi-cell type. So you have fat cells, you have fibroblasts, you have immune cells. But when we look at this particular picture on the right as a sort of depiction of high-grade serous ovarian cancer, why have we been using two-layered, two-dimensional assays to mirror these interactions? It really, for us, again, it went back to understanding the biology and what question we were looking at. Were we trying to understand these intercellular interactions? And if so, then we actually had to design models that looked like the disease biology and would actually help mirror these interactions. So we have organoids where you have different cell types interacting with each other, and then you have organoids where you have the different cell types interacting with each other and their microenvironment. This became important because when you start off with a model that really recapitulates sort of the disease biology, you have a much higher chance of getting compounds into the clinic. So traditionally, we've done a lot of 2D layer 2D monolayer assays, and the gold standard really is clonogenic survival assays, and then you have them going into animal models, which may or may not be the right model for that particular disease, and obviously that results in a high attrition rate of compounds. Furthermore, again, like I said, not every disease has a relevant animal model, and sometimes you in an effort to understand sort of or really in an effort to emphasize the genetic association, we take out diversity from the picture and in our animal models. And then again, you have a high attrition rate of compounds going into the clinic because the patient population is way more heterogeneous than your animal model population. So if, on the other hand, like I said, if we start with more physiologically relevant models, we have a much higher success rate of getting compounds into the clinic because you're starting from the very complex. So you have fewer things to follow up on, but chances are they're going to be efficacious. So I'm going to describe some of the things that we kept sort of in mind with development of high throughput solid tumor models, and then go into a story about a metastatic model. In terms of understanding the intercellular interactions and how they changed as you increase complexity, we ran many solid tumors through a 2D, a 3D spheroid, and 3D organoid models to really see what classes of compounds were active in all of these assays that we were running. And one of the first things that we had to do with organoids is really establish growth characteristics for it and understand, whether this was truly robust and reproducible over time. So when you're used to running 48-hour assays, taking it out to 10 days, 12 days, and 2 weeks, and establishing growth criteria for them became very, very important. So as you can see here, from measured parameters on the Acumen, which is a cytometer, there was great correlation between, plates for both GFP intensity, spherical volume, and then the correlation between these readouts. And I should have said at the outset, this is actually using an ovarian cancer cell line that expresses GFP. So having developed a robust and reproducible assay, we then wanted to look at the multiple parameters that we could use to describe the biology or the sort of the mechanistic effect of different drugs. So what you see here are the HAA8 GFP cells that were also stained with DAPI and PI, and then we have a pancreatic cell line here, sort of just for comparison, to show you that as we look at proliferation, by cell titer growth, which we're normally used to doing, we also started looking at where these proliferating cells were in this organoid and whether we were actually getting cell death over time without any treatment, because that also sort of gives you a sense of what your cell population looks like and where you want to hit them with treatment, because you usually want to look at a system when it's in a logarithmic phase of growth. So what we see here on the left-hand side, you'll see cell proliferation data for selenonomycin and vorinostat, and the profiles look very similar, extremely efficacious with respect to their IC50s. But what I really want to drive home here is that a lack of cell proliferation is not equivalent to cell death. And this is beautifully sort of represented on, in the figure on the right, where you can see that they both trend towards showing almost, completely obliterating proliferation. But the difference between selenonomycin and vorinostat, if you look at that treatment at 50 micromolar, is as well as, a treatment at even 0.4 micromoles, is that selenonomycin completely decimates the organoid. So there really isn't, going back to that, the necrotic zone, the proliferating zone, the quiescent zone, treatment with selenonomycin does not suggest that you will have a population of cells left to repopulate or, to mutate and sort of become a secondary tumor. Whereas when you treat with vorinostat, the entire organoid stays intact, though most of the cells really are dead. So what does that tell us about what is all of that information or the multi-parametric readouts give us for different compounds? What it ends up giving us is a blueprint for different compounds. So not only do you look at efficacy and potency with the readout that we're used to using in screening in a very high throughput system, but you can sort of reduce the throughput, increase physiological relevance for multiple different compounds in different systems, and get a readout for physiological responses that may or may not track with the sort of the high throughput response that we're always used to seeing. And getting sort of looking at these differences in potency and efficacy across multiple assays really underscores the importance for us of taking into consideration various parameters so that you can accurately, or to the best of our ability, accurately sort of develop a blueprint for the mechanism of action of a particular class of drugs or a particular drug. So once we've talked about having complexity, but what we haven't really discussed so far in the development of these reproducible models is, what is the interaction of different cell types within that tumor milieu? So we had we had a partnership with Inspiro, which was sort of a pioneering company, the first actually to develop not only services but products that were marketed entirely for the 3D market. And we decided to study this intercellular interaction with Inspiro. So what we looked at here, as you can see, were the two different ovarian cancer cell lines in multicellular tumor spheroids with NIH3T3 fibroblasts. Now, granted, these were not cancer-associated fibroblasts, and we did follow-up work with cancer-associated fibroblasts. This was just sort of put together to see if we could actually understand the architecture of multicellular type tumor spheroids, number one, and number two was to see if we could develop an assay that had a built-in therapeutic window. Because as I said at the start, with this mindset about investigative toxicology, it's all well and good to get the most efficacious compounds, but if they don't have an acceptable safety profile, they're not going anywhere. So this was one of the first assays where the fibroblasts were engineered with nanoloop borders. So we could differentially track proliferation of both the fibroblast and the total population with cell type equal. And we also did a lot of immunohistochemical staining on these heterotypic sort of organoids as they moved through time. And as you can see, if you look at proliferation, interestingly enough, for both the cell lines, as they sort of grew over time, you see an increasingly proliferative cell population on the outside and a more necrotic core on the inside. So we ran this assay for about 20 drugs that were already approved for use in human beings, looking at potency, efficacy, acute as well as chronic toxicity across the board, in addition to microtissue size. And the idea really was, can you develop a metric based on multiple readouts to talk about whether a drug would be potent and have an acceptable safety? So this is sort of one example of the data that we looked at. And as you can see here, the blue is a decrease in proliferation or a sort of a positive effect of the chemotherapeutic, and the red is sort of chemotherapeutics that were less effective, or at least less effective in that particular sort of time frame. And as you can see across the board, for the HEA-GFP cell lines, as we increased the complexity, the efficacy sort of fell a little bit. And then as you increase complexity over time, the efficacy falls even more. So introducing physiological complexity into these models does decrease by a margin or two what you would expect to see in a 2D system. But it's extremely important for us to understand the temporal effect of these responses between homotypic and heterotypic tissues, because this heterotype is what is being mirrored in a human being, and drug development is going to be based on this response and not necessarily on what we see in the 2D assays. It was interesting for us because the evaluation of the efficacy and potency indicated that heterotypic microtissues will show a decrease in efficacy, although the potencies actually remain pretty similar between homo and heterotypic tissues. So we then sort of moved our mindset a little bit, having understood that you need complexity with the single cell type organoids, and then you need intracellular complexity with the multi-cell type organoids, and you need a temporal response. We then developed an assay with collaborators at the University of Chicago that really was very groundbreaking in the way that the surgical oncologist at the University of Chicago had designed this. As you can see, we've talked about this. Ovarian cancer is extremely aggressive. There's a huge unmet need to prevent metastasis, because once the cells from in the ascites sort of migrate to the omentum, it's extremely difficult to treat high-grade serous ovarian cancer. So Dr. Lengyel at the University of Chicago had an idea. He was a surgical gynecologist, so he would harvest the mesothelial cells and the fibroblasts from his patients during surgery and send them to me. And we had the GFP positive ovarian cancer cells, and we developed an artificial omentum in a 1536 well plate, and then looked for the migration or the lack thereof, because the idea was to look at inhibitors of adhesion and metastasis as it moved through this layer of all of his patients' primary cells. And the beauty of this assay was that every 20 plates was actually run with a different patient's cells, and yet at the end of the primary screen and the confirmatory screen, and I'm going to be showing you some of that data, the compounds were remarkably reproducible. So what it really spoke to was that even so, while we're able to put in the diversity that the patient, or the heterogeneity that the patient population really represents into an in vitro system, the biology is such that only very distinct classes of compounds were implicated in metastasis. And, I think we actually ended up finding a lead compound that is looking very promising that I'm going to be talking to you about. So this was the first 3D high throughput screen that was run with primary patient cells to target unique patient biology. The first 4 compounds that came off of a pilot screen were actually tested by our collaborators in a metastasis model. And as you can see, they significantly reduced metastasis in their mouse models. We then took a 96 well plate assay that they had designed at the University of Chicago and went through the QC process to develop a 384 as well as a 1536 well primary screen. And once we were at a stage where the Z factors were acceptable, we started screening this in a 1536 format. All of the control compounds were equally efficacious. We ended up going with tomatine. And as you can see on the Acumen, this is what the readout would look like. A positive inhibitor of adhesion would look like this because it doesn't allow any of the GFP positive cells to adhere or migrate. And the DMSO control didn't inhibit anything. This was also one of the first high throughput imaging screens for which we had a built-in counter screen for viability with cell titer flow, so that we could also tell whether we had a therapeutic window for anything that was preventing adhesion and metastasis, but not killing the cells, which was extremely important. Everything that we generated from the in vitro screen was then taken by our collaborators at the University of Chicago and beautifully recapitulated in all of their in vitro models. So all of the compounds that were efficacious in one cell line were mirrored beautifully in the other cell lines for high-grade serous ovarian carcinoma. This again becomes important because cell line, there could be cell line differences. Different cell lines reflect different stages in high-grade serous ovarian carcinoma biology, but for metastasis, they all had the same profile. They then took these compounds and when sort of ran in vivo validation assays. And again, the recapitulation of the biology was beautiful. As you can see here, in the in vivo biological assays for adhesion as well as invasion, these compounds were extremely efficacious. And they also, as I sort of mentioned at the beginning, they also prevented in vivo metastasis. A beautiful extrapolation of why this is extremely important, or the role that metastatic disease plays in survival, is mirrored by the data that they sort of produced here, where they looked at the same compounds and looked at survival in these mouse models. And as you can tell, the same compounds that were sort of very efficacious with decreasing metastasis in vivo obviously also was associated with an extremely good sort of survival profile. So what did our screening campaign then look like? Having run sort of a very pharmacologically diverse, active, small library, we then went into a screen that covered over 50,000 compounds. We started off with the Citrovan library, which is approximately, or at least at the time, was approximately 44,000 compounds and was a diverse compound library. It was enriched with very drug-like scaffolds. The impact, on the other hand, at the time, 2,100 compounds, but I believe it's closer to 10,000 now, was a much more biologically diverse library. So the idea really was, all of the mechanisms that could possibly be probed, the impact sort of mirrored those biological mechanisms. We also ran some beta-estin analogs because that was one of the controls, and we ran what at the time was approximately 2,000 compounds, which was our oncology-focused library, a combination of approved and investigational drugs with targeting different cancers. And what we found were that there were many clusters that sort of worked in a similar manner. So we had an incredibly talented informatics scientist, Min Shen, who went through all of the data from the primary and confirmatory screens and then clustered them based not only on the ability to inhibit adhesion and migration, but also sort of giving us that therapeutic window and the effect on cell viability, so that our collaborators could then sort of move forwards with choosing what to follow up on. And as it turns out, there were 3 compounds that were sort of very interesting in terms of follow-up. One was one of our internal compounds. The other one was BP121, and then there was milciclib. And all of these 3 compounds, again, recapitulated beautifully in metastasis and adhesion assays in multiple other cell lines and in their in vivo models. So what we then sort of wanted to do was look at the potential polypharmacology or sort of hitting of multiple targets for both BIP2 as well as milciclib. And what we found was that the first compound, PPI2, it did inhibit mTOR phosphorylation downstream, but then when we tested the same models with other mTOR inhibitors, they didn't seem very efficacious. We had a similar sort of story with milciclib, which hits CDKs and cyclin 4 and 6, and which eventually sort of prevents phosphorylation of the retinoblastoma protein. But again, other compounds in the same class that do the same thing had no effect on migration and adhesion. So we then sort of generated analogs for this lead compound here and found that not only was the lead compound very efficacious in vivo adhesion and metastasis assays, but its 4 SAR analogs were also equally efficacious. And, you know, we've done some further studies now and are actually in the process of writing this up, the pharmacokinetic profile of this compound, and its behavior in ADME studies has been extremely favorable, and there is a good chance that we will be submitting this for a pre-IND pretty soon. So the point I really sort of want to bring home here is that this is work that started in 2013. We're now in 2021, and we're still technically in the discovery phase. But because we started with a much more complex model, the probability that this is going to be an efficacious as well as a safe molecule, and with the potential to have remarkable success in preventing metastasis in human beings is extremely high. So, you know, that brings me sort of to my sort of thoughts that I would like to conclude with. And sort of the one thing I do want to say is that there are no catch-all models. It is extremely important to understand biology and ask the right question. I do think from a regulatory perspective as well, there is a mindset in the discovery space that sort of, you know, it really is moving towards testing efficacy and safety in similar models. So it's not going to be, you know, easy to do this in the same model for everything, but I do think we have to bring in the concept of a therapeutic window or index early in drug development. And last but not least, you know, from the scientific perspective, there is a huge need to recapitulate patient biology in the in vitro environment. It's getting much easier now with functional genomics tools, with iPSC-derived cell lines. So I think as an in vitro community, there is really no excuse for us to not be increasing not only complexity, but also heterogeneity into the in vitro sort of predictive models. And as I said at the very outset, if there's anything you take away from this today, please let it be that drug development truly is transdisciplinary, and without collaboration and teamwork, it's impossible to move the ball forward. So with that, I will stop, and I'm happy to take questions.
Madhu, thank you so much. That was fantastic. You definitely focused on a number of issues that are really important to this group of people, and I'm excited to open the floor for questions. Okay, so I could definitely start.
Sure.
One of the things that you definitely made a strong case for the importance of developing the right model for the right question and adding complexity to reduce the model from being a catch-all, right? And
right.
Without a doubt, this is a really exciting time with I feel like there's so much innovation right now in terms of the models people are developing, the capabilities that they have to develop these models, including the cell lines that are available. I mean, we're just in a really exciting time. However, there's major challenges right now, and you brought this up in your presentation in terms of trying to standardize.
Yes.
Among even you brought up the point of within a lab,
Yes.
and even your specific example was actually just the data, right? But clearly the model and other things are huge barriers. So we have all this innovation. People are exploring the development of these models, but there are definitely major challenges in trying to develop them to the point where they can be validated, standardized, and disseminated. So that I kind of in the introduction had mentioned that you established like a working group of people in different sectors to kind of focus on these issues. What do you think right here, at this point are the best, kind of some of the low-hanging fruits that could help people in doing this? Important, validation tools or reagents that, that could help people, or even data analysis methods so that people could better standardize what they're doing.
Absolutely. And so you bring up, you expanded upon something that's extremely relevant to the community, right? And this is something we've struggled with over time, because the first couple years was people coming together to accept that we actually have to do this, right? We're all developed, and as you have newer plates and newer models and newer imaging techniques, the data just becomes more and more complex and more nested, right? It's interesting that you bring up this innovation versus standardization mindset, because there are labs that worry too much that standardization will squash innovation, if that makes sense. If you have to worry too much about the standards, you won't innovate. But I would almost argue that we've already innovated, right? But as we innovate, if we want the models to mean anything, then we have to standardize it along the way. And what's very heartening for me to see is regulatory agencies being willing to engage in this early discovery space so that they can really understand, data that's being submitted for, for pre-INDs or INDs. Typically, we know what animal model data is supposed to look like. But if we have these predictive models, and people are willing to share their data so that the so what of the regulatory unmet need can be understood. I think people coming together in the pre-competitive space to share data that they use internally to make go and no-go decisions becomes important, because then you can compare across the board. I think right now we compare our data within the, among the rest of our data for a particular lab. But what that body of data looks like compared to another lab, is a tough call. And I think, with regulatory agencies wanting to share more data about adverse events and, how you can mirror that in vitro models, if we want to make a one-to-one connection, we do have to have the standardization at the in vitro level, right? And you brought up a good point. Where do you begin with standardization, right? Because there's technical standardization of just the, how cells grow in a plate. There's the data standardization. What does it mean? Was anything wrong with a particular batch? And then if a particular batch looks different from another batch of experiments, is there a way to look at the data in the context of that experiment so you don't have to throw it away? So you have to understand a lot more about, how you normalize within controls and how your controls behave from batch to batch. So I think if we were able to break up technical standardization and then biological characterization of these assays separately, and then just go step by step, I think breaking it down into bite-sized pieces makes it more amenable and, more doable. And then as you see successes in each one of these areas, I think we will eventually, hopefully, get to a point where there are set standards for set readouts for set biologies, and we can use that as to build up from there.
Do others have questions for Dr. Lal-Nag?
Hi, that was a great talk. Really enjoyed it. The last thing you left us with, the idea of using the same model, the same preclinical model for safety and efficacy is really, it's really intriguing. Is there a precedent for that at this point?
No, Dr. Friedman, there isn't. And I think that's the issue, and I'm sure, Dr. Cousins can speak to this as well, as can many people in the audience. We've made so much progress with developing models for efficacy, and yet we're looking at safety profiles in Hep G2 cells, in HU8 cells and CACO cells, and it just doesn't represent the heterogeneity and doesn't represent the disease biology, of a recipient cell. And what we're looking at even now from a regulatory perspective is not necessarily only the drug, but its component metabolites in a disease tissue, and whether we should be testing that as well, because oxygen gradients and pH gradients are different in a disease tissue than it is in normal cells. So very few people, to answer your question, very few people are looking at this, so there isn't a precedence to do it. But I think that some people, there's a predictive safety testing consortium with the Critical Path Institute that brings people together in this space. There's IQMPS. I think there are groups of people that are beginning to talk about this. And I think if they're able to get enough consensus, this could be integrated into that whole data sharing part, because I do think it's important.
Thank you. Thanks.
Do others have any questions? Yeah, Madhu, I mean, you were even bringing up the whole additional layers within just a disease that you're trying to model in vitro. Depending on when you're looking at this disease, it can be radically different. And so really you have to design your model system around that particular stage to hope to have a more, I guess, a model that's not as much of a catch-all.
Right.
To help you find better what you're looking at.
Right.
And that's just a very challenging thing to do with cancer.
Absolutely. I mean, and one area that and obviously this is very oncology focused, it's extremely challenging with cancer. And it's also extremely challenging in sort of looking at different demographics, like, a pediatric population versus an adult population. It's funny because we intuitively understand that those two populations are very different when it comes to therapeutic development, but it's almost an extrapolation of the same mindset. Someone who's suffering from stage 1 versus stage 3, HER2 positive breast cancer, it's actually two different diseases, and I think if we were able to look at it in that sense, we would be, it would be, I hate using the word easier because it's easy to say, but I think it would change the mindset about developing our models.
Another thing that struck me about the presentation you gave is, thinking about the continuum of more complex models, right? At some point, you get sort of to the edge of the in vitro models, and then you're into, animal models.
That's
and animal models, I mean, at least, when you're thinking about tumor models, you're looking at tumor growth over time, right?
Yes.
And it's when you're thinking about the more cell-based assays, people are typically looking at, cell death or cell viability at a given time point, and sometimes just a given concentration of a test compound. So those are kind of, just different ways of looking at, things that are not necessarily aligned. So I was pretty intrigued with your approach of, looking at spheroid size over time. Is it shrinking? Is it growing? Do you find that that's a better, do you have any evidence that suggests that correlates better with animal experiments?
Yeah, well, excellent question, because again, that's yes and no, right? Even with animal models, sort of going back to my postdoctoral days, it depends on how you implant a tumor into an animal, right? Is it a sort of a xenograft, orthotopic? Is it a genetic model? The disease manifests very differently. And I think that's what we're trying to get to the crux of. When you implant, sub-Q, a sort of set of sort of cancer cells into a mouse, is that really the right model? It has to grow in its environment to be able to recapitulate that. So I'll give what we saw with the mouse models for metastasis were remarkably similar to the in vitro system, but I think we haven't quite delineated the same. What's the word I want to use? Maybe extrapolation for solid tumor models. When you look at hematological malignancies, again, because they're usually so genetically driven, there's a good sort of correlation between your screening or the in vitro models and what you actually see in a mouse. So I think you ask a very important question. I just think that we haven't established that correlation across the board. And to be honest, I don't know that we will be able to, because I think the bigger lesson here is not that we're trying to replace animal models yet. I think we're trying to get to as predictive as animal models. And then what can you do to make it better than an animal model, right?
Mm-hmm. Yeah. Any other questions?
Maybe I can try asking a question. It seems to me that based on your talk, the idea is really to almost do like I'll use the analogy like personalized medicine, maybe personalized screening to certain disease states and let's say the way of designing the assay to make it amenable to a certain disease state. I feel like as a screener, one of the issues that we come across is trying to design a screen that would maybe not be a catch-all, but that would try to encompass as many disease states as possible. And I didn't know what was the best way to move forward. Isn't that to, let's say, miss as fewer hits as possible and then deconvolute later? Because if we try to follow, let's say, the path that you're taking, trying to be too specialized might make you miss some hits that might be relevant in some other areas.
Absolutely. And that's where we go back to talking about, do we know what question we're asking before we start screening? So I completely agree with you. I don't think you want to start with an organoid model with built-in flow and tension, etc. But I think it comes back to asking, what is the underlying mechanism that you want to look at for a particular disease state? So, for example, if for breast cancer, you're looking at how does it go from a carcinoma in situ to migrating into the sort of having lymphatic involvement. What you're targeting right there is breaking through the ducts into and then moving into the lymph nodes. So whatever your assay is, it would mirror not necessarily the organoid that would that you would have to build in complexity for the primary breast tumor, but the cellular interactions between the lymphatic milieu, for example, and the migrating cells. So I think I completely agree with you. You don't want to start with too complex. And perhaps one of the things I didn't expand upon this, but I think when I showed the continuum, I think the idea really is you start off with a broader question and a simpler assay, right? But what you do know is that whatever that broad question is, if the compounds are not hitting that biology or hitting that, or not answering the question there, then they're not of interest for you in this disease context, right? And then you take the same set of compounds that were active in the much broader question and you increase complexity for the next layer of biology that you're looking at. And you sort of take a look and say, the compounds that are now coming are positive in 1 and 2. How do they relate to the next question that I want to ask about this particular disease? And I'll give you an example. I couldn't go through the whole protocol for the metastasis assay, but actually there was a whole lot of many ways that we looked at that data. So to your point about how much complexity do you want built in, we looked at compounds that were inhibiting adhesion at 16 hours, which had a good therapeutic window, but at 16 hours that may not have had too much of a therapeutic window. Then we did the same at 48 hours. And what I showed you were the compounds that went through from a 48-hour inhibition of metastasis. So you're looking at a chronic endpoint, but there were other endpoints from that same assay for metastasis that we looked at that spoke to different components of what metastasis and high-grade serous ovarian carcinoma looks like. And we sort of gated those compounds based on the questions that we were asking. So I think you're absolutely right. You can't start off with too complex, but as long as you know for the simpler assays, what that means in the context of the more complicated questions you want to ask later on, I think you can build in complexity into assays, but always have sort of that bigger sort of web of compounds that were active in your disease biology. Does that make sense? Did I answer your question?
Yeah, thank you for the explanation.
Okay.
So indeed, you want to start with a broader assay and possibly have orthogonal assays, or even
Correct.
have
Yes.
multiple endpoints and make your
Absolutely.
judgment based on those
Yes.
endpoints.
Yeah. Yeah. Absolutely.
Okay.
Because I do think they nest into each other, or they should be made to nest into each other. But you can't answer disease biology by starting off with something much more complex, because you don't know what sort of this side looks like, in terms of the mechanisms.
Sure. Thank you.
Thank you.
So Madhu, in terms of, I know one of the interests you've had, for longstanding really, is to provide more reference standards in
Yes.
the community of people that are using tumor models. What do you think are the biggest needs there for the field right now?
That's an excellent question. In fact, I'm with some colleagues working on a position paper right now on the technical standardization and the need thereof. I think that when you talk about something like biomarkers, you talk about qualification and you talk about validation. But we don't necessarily do that with tools, right? And at the end of the day, we need standardization tools for all of the data that we develop to be able to move it to the next step. So I think right now, I would say technical standardization from an assay perspective, but also data standardization, because we produce a lot of data, and I'll say something that may not be very popular, but I don't think we always know what our data means, right? And I think that is extremely important. Till you understand what your data means, not only in the context of the assay, but in context of sort of the bigger biological question, it's hard to sort of make any conclusion from any assay. So I would say the needs right now are data governance, data normalization, how we collect data, and how that sort of fits together.
Yeah, that definitely resonates with me as being an important problem. We find that there's a variety of perfectly appropriate ways to analyze data, and you can get different metrics, and they're not necessarily all aligned. Like, for example, an IC50, an area under the curve. So if you're looking for correlations, if you're trying to think about from this particular readout of this model, what's the most important thing that I should be paying attention to? It's very difficult. Same's true with the in vivo models too.
Right. Absolutely. Absolutely.